Picture a dim lab at midnight. The instruments hum like a distant city. You, a curious researcher, hold three keys: breast, ejaculation, and the mysterious nocturnal orgasm. Each key opens a door to biology, culture, and unanswered questions. Listen quick, there is a map here for experiments and ideas that will make you want to read every paper and design new studies tomorrow. First door, breast, not just tissue and fat, but a patchwork of milk duct, gland, and a sensitive island of nerves in the nipple and areola. They are built for feeding, shaped by hormones, estrogen and progesterone in development, prolactin and oxytocin for milk. And around the world, they carry heavy cultural meaning. From the lab view, breasts are a perfect model of dual function, reproductive and social. Simple experiments can tease apart touch pathways that feed into sexual arousal versus those that trigger milk ejection. How does oxytocin act differently when the brain calls for bonding versus when it triggers lactation? Can functional imaging map the change in brain networks from sexual touch to caregiving? Sensory mapping, nerve blocks, hormonal probes. Each method is a clean tool to answer messy, fascinating questions. Second door, ejaculation. Think of it as coordinated choreography between peripheral organs and the spinal cord. There are two main acts: emission, where seminal fluid mixes in the reproductive tract, and expulsion,
the reflexive motor burst.
The sympathetic and somatic systems take turns leading. Semen itself is an ecological mix. Spermatozoa are carried in seminal plasma with proteins from the prostate and seminal vesicle. Each component has a job. Protect sperm, modulate the female tract, signal fertility. For researchers, ejaculation is a window into evolution, immunology, and neurobiology. Which molecules in seminal plasma influence the partner's immune response? What neural circuits translate genital sensation to the burst of reflexive muscle contraction? Modern tools, genetics, proteomics, spinal imaging can map these steps without sensationalism and reveal adaptive stories written in molecule. Third door, nocturnal orgasm, often called sleep orgasms, or the lesser studied cousin of nocturnal penile tumescence. These events blur sleep. They happen in REM-rich nights when the brain runs internal simulation. Hormones shift across sleep cycles, cortisol dips, growth hormone spikes, and sex steroids follow slow rhythm. The result, spontaneous sexual arousal, and sometimes orgasm, without conscious intent. Prevalence data are sparse and biased by self-report. Objective study needs polysomnography, heart rate, and muscle sensors, maybe non-invasive hormone sample. Imagine a study that pairs sleep EEG with genital airflow and salivating oxytocin spikes. You might finally know whether nocturnal orgasms are triggered by dream content, by autonomic overflow in REM, or by hormonal pulses. Across all three doors runs an ethical thread. Consent, privacy, and cultural sensitivity must shape methods. Wearables, anonymized data, and clear participant information let us study intimate events responsibly. There are low-hanging fruits, cross-species comparisons of mammary signaling, Proteomics studies of seminal fluid across populations, sleep lab protocols that quantify nocturnal orgasm frequency and neural patterns. So walk out of the lab at dawn with a list. Map nipple brain circuits, characterize seminal plasma signal, measure REM-linked autonomic bursts. Each project is simple, doable, and rich in discovery. These topics are not just titillating words. They are a living system where hormones, nerves, behavior, and culture meet. Open the doors, design careful studies, and let curiosity lead the way.